चलेगा ना? ओके। सर्व सुविधा जरूर सुरू
हेलो कौशिक जी।
यस सर।
हाँ, पुणे का हाइट बढ़ा गया ना, उन्होंने बस
हाँ सर बढ़ा दे सर। हाँ सर सर ने बोला था थोड़ा ऊंचा कर देने के बिकॉज पीछे इतना केस के सारे देख पाएंगे डॉक्टर्स वगैरह।
परफेक्ट परफेक्ट, ठीक है।
ओके सर ओके सर। रांची भी सही है, अभी कलकत्ता भी सही है। कलकत्ता को और थोड़ा जूम करूँ क्या? रौनक सर अपना साउंड नहीं आ रहा।
हाँ, मैं बोल रहा हूँ, एक चीज एंश्योर कीजिए, ऑल टेक्नीशियंस को बताइए, द मोमेंट नेशनल मॉनिटर विल स्टार्ट हिज टॉक, सो सबकी स्क्रीन पे ही शुड बी विजिबल एट ऑल लोकेशंस।
हाँ तो सर सबको विजुअल आ रहा है।
हाँ, second is when the doctor Professor Christopher Gash starts his presentation, so PPT should be visible at all screen, no side windows. So full screen view they have to do it.
ठीक है ठीक
Manually
है
at each location.
सर। मैं जोराट भी नजर आ रहा है, तो जोराट की बंद कर दूँ।
हाँ, जोराट का close at side view.
ओके जोराट, हेलो जोराट। जोराट प्लीज हाइड योर स्मॉल विंडोज। यस यस डन।
हाँ, perfect.
डन परफेक्ट। कलकत्ता प्लीज आप थोड़ा बैकग्राउंड दिखाओगे मेरे को?
रांची थोड़ा जूम नहीं करना, रांची का वीडियो एंगल बहुत ज्यादा पीछे से है।
हाँ हाँ रांची प्लीज कैमरा थोड़ा जूम कर दो।
उसको बोलो पोडियम पे फोकस करेगा, थोड़ा अपर्चर बढ़ाएगा, लाइट्स थोड़ी उसको बोलो इन हाउस लाइटें बढ़वाए। एट ऑल लोकेशन वी नीड टू
बस बस ओके ओके
दिस इज परफेक्ट।
లైట్ అయ్యి ఒకసారి 1 by 1 చెక్ చేసుకుంటున్నారు, ఒకసారి లైట్ వేస్తే
ठीक है
ओके सर।
पैरेलल वाले को भी बोलो थोड़ा सा स्लाइड लेफ्ट करेगा, पैरेलल कैमरा एंगल अपना
लेफ्ट
स्लाइड लेफ्ट
इधर सर,
स्लाइड थोड़ा सा और
ఇంకా లెఫ్ట్ అంటే మనకు లెఫ్టా వాళ్ళు మన లెఫ్ట్
प्रतीक सर,
हाँ जी बोलिए।
you want sort of video
45
what time you kept? 45.
8:30 था नाउ, आपने 8:45 बोला था, तो जस्ट आफ्टर 2 minutes इट विल बी स्टार्टेड।
Fine.
బ్యానర్ లిమిట్ అంటే కట్ ఓకే పర్ఫెక్ట్।
रामनाथ जी
ఇప్పుడు స్టార్ట్ అవుతుంది చెప్తా
रामनाथ जी
స్టార్ట్ 2 minutesలో స్టార్ట్ చేస్తారు। ఇది ఉంటుందా 1 hour? అది పెడతా పెట్టేసేయండి ఇప్పుడే।
रामनाथ जी
हाँ जी प्रतीक सर।
हाँ मैं आपसे पूछ रहा था क्या नाम है डॉक्टर मंगेश सर ने ज्वाइन कर लिया क्या?
No, we are waiting for him. Dr. Shaw?
No, he is not yet joined. He'll be joining in some time.
సైడ్కే కదా
Just have a call.
ఉంది,
तो
ఉంది బ్యాక్ ఫుల్ ఉంది, ఒక్కటే పాయింట్ తగ్గింది 90% ఉన్నారు।
सीक्वेंस वही है,
90%
डॉक्टर मंगेश सर आएंगे पहले, देन डॉक्टर ग्रेस आएंगे, ठीक है? तो अकॉर्डिंगली स्लाइड विल बी रेडी, ठीक है ना?
ఒకసారి లైట్ ఆఫ్ చేయరా?
चलो
You have that presentation also, right?
we have sir, but we kept it as a backup.
ఇప్పుడు
Dr. Chris is already showcasing their screen, so उन्हीं के स्क्रीन से presentation चलेगा।
I would prefer if you have already uploaded Dr. Mangesh Srivastava's presentation.
అంతా రికార్డ్ చేయాలి.
Yeah,
ఇక్కడ
at the backup, I
టోటల్
think we should keep
ఇదంతా
it ready.
తీసుకుంటా
backup is there.
స్టార్ట్
Backup we
చేసుకుంటాం.
have.
Yeah, because if we connect, then there will be a very short time before we go live. So if at all required, then we can just go through on the screen and we
కానీ
can
అక్కడ డిస్ప్లే
continue.
కూడా కావాలి.
డిస్ప్లే వీలైనంత ఒక్క టేబుల్ తీసుకోవాలి.
And few more people are still waiting to join in the meeting.
Like sir,
ఇది నువ్వు ఎప్పుడు అంటే అప్పుడు
you name them, will join them.
I think Rajni is still
యూట్యూబ్లో
waiting.
పోతుంది
Rajni is still waiting. Arey,
యూట్యూబ్లో పంపిస్తారు.
మమ్మల్ని ఎక్కడికైనా ఇది కావాలనుకుంటే రికార్డింగ్ కొట్టాలి.
ఇప్పుడు ఇది కూడా రికార్డ్ చేయాలి సార్. సార్ ఇప్పుడు ఇది రికార్డ్ చేయాలి, ఇది రికార్డ్ చేయాలి, అది రికార్డ్
just
చేయాలి.
నేను పెట్టేది రికార్డ్ చేయాలి.
ఇప్పుడు మీరు పెట్టింది ఇది రికార్డ్ చేయాలి, ఇంకొక వీడియో కెమెరా కూడా రికార్డ్ చేయాలి. ఇది ఇట్లనే ఉంచండి స్క్రీన్, ఇంకో కెమెరా సార్ వాళ్ళు ఎటు మూవ్ అవుతున్నారో అటు మొత్తం తీసుకుంటారు. స్టార్ట్ రెడీ చేసుకోండి. 2 minutes స్టార్ట్ అవుతుంది.
ఓకే.
దానికి
రికార్డ్ చేయాలన్నా వద్దా నాకు ఐడియా లేదు. బట్ ఇది రికార్డ్ చేయాలన్నా వద్దా అవుతుంది. అది ఆయన అడుగు, మీ కౌశిక్ అడుగు, ఇది రికార్డ్ చేయాలన్నా వద్దా అని నాకు తెలియదు.
1 hour
ఇదైతే రికార్డ్ చేయాలి.
ఇదైతే సెకండ్ కెమెరా కంప్లీట్ రికార్డ్ చేయాలి.
అది ఇది తెలియదు, నాకు ఐడియా లేదు.
మరి నీకు ఆడియో కావాలి, దానికి కూడా కేబుల్ వేయాలి, ఇంకోటి ఆడియో
దానికి డిస్టర్బెన్స్ అయిపోయింది.
వాళ్ళు డౌట్లు అడిగినప్పుడు మైక్ ఉందా, కలర్ మైక్?
మైక్స్ ఉన్నాయి, హ్యాండ్ మైక్స్ ఉన్నాయి, ఎక్కడంటే అక్కడ అడగచ్చు.
जयपुर कैमरा थोड़ा सा
ఉంటుంది.
जूम आउट करो.
సరిపోతుంది కదా మీకు?
సరిపోతుంది.
మళ్ళీ చెక్ చేస్తున్నాం జైపూర్.
ఇది ఎప్పుడు వస్తే అక్కడ పోతుంటాం.
ఇదైతే రెడీ పెట్టుకోండి, ఇంకో కెమెరా
రెడీ.
but this organization says sound and close because at 45 people are welcoming at respective locations.
So all muted है, all locations are
And sound भी mute कर दिए. Sound.
sure.
They should not be audible.
So Atesh, please do this.
జస్ట్ నేను ఆఫ్ చేసి పెట్టాను.
Now done, sir.
ఎందుకంటే ఇప్పుడు ఈ స్పీకర్లో వచ్చే వాయిస్ అనేది మళ్ళీ అక్కడ వెళ్తుంది. అబ్బాయి మీరు జస్ట్ ఆన్ చేసుకొని మాట్లాడండి. ట్యాబ్లెట్ తెచ్చినా బాబు లేలే ఒక రెండు సిట్రజిన్ 2 కోల్డ్ యాక్టివ్స్ సిట్రజిన్ 2 కోల్డ్ యాక్టివ్స్ అయితే 4 తీసుకురా ఒక 4 సిట్రజిన్ 4 కోల్డ్ యాక్టివ్
Dr. Sanjay?
Yeah, Ramnath.
Yeah, Dr. Mangesh is not joining here.
Yeah, I've already messaged him again.
That's fine, call no.
Yeah.
ఇది అప్లోడ్ చేద్దామా డైరెక్ట్ యూట్యూబ్లో కొడతా అవుతుందా అవుతుంది డైరెక్ట్ లింక్ ఇచ్చేస్తే అయిపోతుంది సాఫ్ట్ లింక్ ఇచ్చేస్తే అయిపోతుంది
He's just joining, Ramnath.
Okay, no.
Yeah, he has received the presentation in this.
So he's be sharing from his side.
Good evening, respected doctors. It gives me immense pleasure to extend warm welcome to all of you on behalf of Aristo Pharma, MF3 division. Today, Aristo Pharma is the 10th largest by value, 5th largest by prescription company in Indian pharma market. At Aristo Pharma, we believe in building concepts through science. First time in India, we launched US FDA approved oral ferric maltol with the brand name of Megaferon. And with your support today, we are the #1 prescribed brand of ferric maltol. Ferric maltol is a unique molecule compared to the conventional iron. As you all know, medicine is a constantly evolving field, and staying updated with a newly researched molecule like ferric maltol is the need of the hour. To learn more about the molecule, we are privileged to have with us key researcher molecule Dr. Christopher Gashe from Vienna, Austria, and Dr. Mangesh Tivaskar from Mumbai, and Chandrasekhar sir from Warangal, regional moderator, will guide us through today's session. Their insights and experiences will surely broaden our understanding and help us translate this knowledge into the better clinical outcomes. I once again welcome each one of you. Let us make this CME a meaningful and enriching experience together. Now we all start online session in few minutes. Thank you.
Kaushik ji,
Yes, sir. Yes.
YouTube streaming.
So YouTube streaming. Adesh, can we start the YouTube streaming, please?
Yes.
Already started, sir.
नहीं दिख रहा है.
Adesh, एक बार चेक करो.
లింక్ షేర్ చేస్తాను ఆల్రెడీ ఇక్కడ అయితే వేరే వాళ్ళు చేస్తున్నారు
క్లోజ్ అక్కడే క్లోజ్ కొట్టాలి.
Hello. Good evening, Dr. Devaskar. I can't see him. Dr. Devaskar.
Yeah, keep the permissions on for muting and unmuting and don't stop it. I'm sorry, I was stuck in the traffic.
Yeah.
So I could join a little late. Am I audible now?
Yes, sir. Now you are audible.
Yeah.
So I have sent you the tentative script as you can go through it. And there are some 6 locations already connected with, and they will be having local operators. Their name and brief credentials I have included in the script also. And in the slides also. So,
perfect.
Audience
and besides that, there is one 7th center at Kolkata, where there may not, there will not so you will be sharing your screen with the presentation, or
You're asking me or sir?
yes sir, yes sir.
No, it's okay. In fact, we can be just like that if it is okay, all right for you.
Ramnath, if you have the backup ready, then we will start with that.
Presentation.
Yes, Pradeep sir.
Just a person chinna block lagna banja.
Give me a card so that we will be ready. हाँ जी सर बताइए.
Ramnath, you are on mute.
हेलो प्रतीक सर.
हाँ जी सर बताइए.
हाँ, डॉक्टर मंगेश का पीपीटी जरा शेयर कीजिए.
या अपडेट.
His
Or if
presentation
Dr. Devaskar is okay to share from his system, that also would be fine.
Sir, totally.
That is,
No issues at all. I'll share.
yes, that would be better.
I'll share. Don't worry.
Okay.
Just give thumbs up, Pratik sir, when you are about to go live.
Dr. Grace is there?
Yes, I'm here.
Yeah, yes sir. Yeah, hi. Yeah. So there is an introduction presentation, and that includes the 6 slides of the 6 local moderators also.
Okay.
Dr. May I introduce you to Dr. Mangesh Devaskar. Sir, Professor Chris is here from the University of Vienna.
Yeah.
And Dr. Mangesh Devaskar will be the moderator for the today's session, sir.
Yeah. I think Sanjay, the presentation that we have shared does not have that the other moderators' details. So I would request you guys to share that presentation straight so that it will be
Yeah, this is the old one that you are. Today I sent you today only that presentation.
Asha, you have WhatsApp me or you have emailed me?
Both email as well as WhatsApp.
Okay, so I'll check that.
Yeah.
I request that it would be better if you can share it from the presentation side so that there is no confusion. Okay.
Yeah, sure.
Okay.
Hello. Yeah. Ramnath. Ramnath, you are on mute.
Yes, sir, I'll share from my side.
Yeah, and sir would like that new presentation with the details of 6 local moderators and all to be flashed here.
Yes.
Perfect.
Yeah, he has that earlier presentation without the details of those 6 local moderators.
Fine, I will share those.
Yeah.
But Chris, we were there last month in Vienna for an ESD conference. We really loved that place.
Last
Yes.
month in November?
Yeah, just end of October and November. There was a international European Society of Diabetes conference, ESD, that happened at Vienna Convention Center. So we were there.
I hope you enjoyed it.
Yeah, really it was beautiful.
Yeah, my screen is visible?
Yes.
Yes. Pratik sir, are we live?
So give us a countdown so that we will go.
You just change the slides and see once.
Yes, sir, we are live. Perfect.
Yes.
లేదు జస్ట్ ఇవి ఆఫ్ చేయకండి ఆఫ్ చేయలేదు ఇది కొద్దిగా రింకిల్స్ కనబడతాయి
Yeah, Sanjay, we are live, so probably we will
రింకిల్స్
start.
కనబడతాయి కొద్దిగా ఇక్కడ
So the slide has been set to you. You should welcome the delegates.
Yeah, so shall I start?
Yes.
If you want to.
Welcome, sir.
Yeah. Yeah?
ఇక్కడికి వస్తే తగ్గుతాను
Okay, so are we good to go live?
మళ్ళీ సార్ మాట్లాడే ప్రాబ్లం ఎవరు రావాలంటే
Good evening, everybody. I am Mangesh Srivastava.
We are live now, sir.
Yes, so good evening, everybody. I am Mangesh Srivastava. And
కనబడతాను
welcome to today's very, very unique
ఇది కొద్దిగా డౌన్ చేస్తాను
webinar, I would say, international speaker program on the new perspectives on the spheric maltol. Now we have heard different combinations, we have used different iron therapies to manage anemia. But let me tell you that this spheric maltol is one of the kinds that has really impacted the way we manage anemia, especially iron deficiency anemia. We do have here with us today one of the legends in the iron therapy management. He is Professor Christoph Gash. I hope I pronounced his surname correctly. Christoph, sir, is one of the legends who has really published his original work in this molecule. Now, to welcome all of you, let me also tell Dr. Christoph that we have been joined today from 7 different locations all over India from our country. It is Behrampur from Odisha, Pune, Warangal, Jorat, Jaipur, Ranchi, and Kolkata. So thank you for being there with all of us. Now, if you really look at today's session, we will be trying to focus on our relevant daily clinical management calls that we want to hear and learn from you on iron and inflammatory disorders. And in that perspective, what are the new things on ferric maltol? Now, especially in India, if you realize, iron deficiency anemia is extremely prevalent. In fact, that is one of the most commonest illness which is neglected all over India. And especially when it comes to women, almost 8 or 9 out of 10 women are anemic. And the commonest cause is iron deficiency, we all know. And generally these diseases, iron deficiency exists with a lot of chronic inflammatory conditions. And they impact the patient outcomes that we are all aware about it. Commonly presenting with fatigue, reduced functional capacity, decline in the quality of life. All those things associated with inflammatory conditions like irritable bowel disease or the inflammatory bowel disease, chronic systemic illnesses, including the chronic non-communicable diseases like diabetes, hypertension, and also very active inflammatory diseases. They all have some role to play when it comes to iron metabolism and iron deficiency. So we are sort of trying to evolve our understanding about the inflammation-driven changes related to iron metabolism and what are the new therapeutic options which are emerging like ferric maltol. So to address this, I am greatly honored to introduce today's one of the champions and the esteemed international speaker, Professor Christoph Gash. director of Loha for Life. You know, Loha is again a Sanskrit word for iron. In Indian culture, we use it in Hindi, Marathi, and a lot of regional languages also. Loha in Sanskrit is iron, and I am more than sure that the name for his center, Loha, has been inspired from Sanskrit. The Center of Gastroenterology and Iron Deficiency and Management, Vienna. He's a professor at Medical University of Vienna, Austria. He is a board certified clinician, an internal medicine champion, and also he is a board certified gastroenterologist. And he has completed his training in the molecular oncology at University of California, San Diego. Friends, Professor Gash is globally recognized for his groundbreaking work in iron deficiency anemia and gastrointestinal disorders. His contributions include classification of Crohn's disease, his major achievements in the inflammatory bowel disease, immunogenetics, inflammation-associated colorectal cancer, and chemo prevention attracted the global audience to hear him. He has more than 100 international publications. He has written 6 books, 27 chapters in different textbooks, and he owns 4 patents. Now that is remarkable. He is extremely well heard clinician across, and we all are extremely privileged to have him with us today. So friends, I am Dr. Mangesh Tivaskar. I am a physician diabetologist at Shilpa Medical Research Center in Mumbai. I also am lucky enough to shoulder the responsibility of being the editor-in-chief of Journal of Association of Physicians of India, that is JAPI, which is one of the most highly well-read journals in the Indian subcontinent. This education partnership comes with Aristo Pharma, so thank you very much Aristo Pharma for giving us this opportunity to hear from one of the legends of the subject. And together we have Sanjay Shroff from our management team from Medica Press and his team to actually conduct this webinar. Quickly, before we start, Dr. Gash, I'll just introduce the moderators from our local centers. We have Dr. Deepak Kumar Pushti, a general surgeon over 30 years from Behrampur, Odisha. Dr. Dhanoba K. Bhaskar, he is a consultant physician and diabetologist from Pune, Maharashtra. Dr. V. Chandrasekhar, Director of Medical Education and Superintendent at GGH Mulugu Hospital from Warangal. Dr. Sujoy Barua, Consultant Gynecologist and a Laparoscopic Surgeon from Jorhat, Assam. Dr. Madhulika Agrawal, she is a Senior Consultant in Obstetric Gynecology from Jaipur, Rajasthan. And Dr. Beauty Banerjee, she is a General Surgeon from Ranchi, Jharkhand, and I would take care of a Kolkata center. So handing it over to Professor Christoph Gash, I invite him to take us through his talk on iron and inflammatory disorders, new perspective on ferric maltol. Over to you, Professor Gash. Thank you very much. Stage is all yours.
Dr. Mangesh, thank you very much for introducing me. I'm not so important, but I'm trying to advance the field of iron deficiency in gastroenterology since I'm very young. And I will show you a couple of very old slides, very old work together with the latest work on iron maltol, which is the topic of today. I became interested in iron through inflammatory bowel diseases, and this is what I'm going to show you in the next couple of minutes. I will talk about iron deficiency in IBD. I will give you some insights into our IBD mouse models. I will a little bit go into alternative iron products. Iron EDTA is part of food supplementation, and I will give you some data on that in comparison to ferric maltol. And then I will end up with the clinical IBD trials on ferric maltol in IBD-associated anemia, the so-called AGIS trials, and I will end up with showing you the safety profile of this product that really changes IBD iron management around the world.
So I might say that, was I right to say that, you know, your name of the center, Loha, is being inspired by the word Sanskrit Loha, that means iron force?
That is very correct, and I will point you to the person who invented this name at the very end of my talk. Yeah, I have a co-worker for many, many years, and she's from India, and she came up with that name. So yes, you're perfectly right. That is correct. Lower means iron. IBD has a lot of complications, and the most extraintestinal complication is anemia. Anemia is in IBD is multifactorial.
Professor Kisov, sorry to interrupt. Your screen is not visible. You have to share your screen.
You need to. Your slides are not shared, sir. I believe. So you have to share your slides.
Once again, yeah, please.
Okay, let me try to do that. I did share my slides before, but I'm trying to share it again. I'm sorry. Can you see it now?
Yeah, they're coming up, sir. Yeah, now we are able to see it. Yeah.
Okay, I'm sorry.
Very fine, sir.
Thank you. I was introducing you to the structure of my talk about IBD, IBD mouse models, and about clinical trials with ferric maltol. And I was talking about IBD itself as anemia itself as the most common extraintestinal complication of IBD. And the main cause of anemia in IBD, and we'll go into detail about this, is iron deficiency. This is basically studies out of the late 80s and early 90s which we conducted. The second cause is inflammation, and I will go a little bit into the molecular details of that. Of course, there is also nutritional deficiencies. I guess in India as well as in Austria or Europe or in the United States. We use also medications that impair the bone marrow and can cause anemia, such as azathioprine. And there is a rare causes of anemia, such as hemolytic anemia, but these are the rare causes of anemia in IBD. Anemia is associated with disease activity. Everybody thinks because it's associated with disease activity, this is due to the inflammation. No, it's mostly because disease activity comes with ulceration, and ulceration is seen on the right-hand side. Typical ulcerations in Crohn's disease below, and ulcerations in ulcerative colitis. I don't need to show this to you. Everybody knows these pictures from endoscopy. And these ulcerations are causing bleeding, and bleeding is causing iron deficiency. And anemia itself is highly neglected as a disease, as it is so common. You're mentioning iron deficiency being very common in women in India, and they are probably used to this iron deficiency anemia, but it adversely affects quality of life. You mentioned fatigue as one of the most important symptoms. And once more, I will show you that anemia is recurrent. It's not a single one stop that you need to treat. It's a continuous issue that you need to take care of your patients. So this is where everything started off, and I will point you. I'm not sure whether you can see my error, but this is here in 1994, published in 1994 in Digestive Diseases and Sciences. This was basically the first work I did in anemia. On the cause of anemia. And the question was, is it more iron deficiency or is it more inflammation? And we studied about 450 patients with IBD, exactly 49. 16 were anemic and 33 were non-anemic. And we compared, the clinical activity, the C-reactive protein, other inflammatory markers like alpha-1 acid glycoprotein. And we studied iron parameters and erythropoietin. And we came up that the most important issue here that was significantly different was ferritin. Ferritin was significantly lower in patients with anemia compared to patients with no anemia. Anemia also comes with erythropoietin increase. There was an erythropoietin increase, but we considered this increase inadequate to the severity of anemia. I also mentioned that anemia is very prevalent. The studies are varying between 36 and 90% in the IBD population. And I also mentioned before that blood loss from the ulcerated mucosa is the main cause of iron deficiency. However, there is also reduced uptake, and this is important when we think about oral iron therapy. And there is also impairment of the uptake. And the most important issue is that there is a persistent negative iron balance, which means that you're losing more iron over the ulcerated surface of the intestinal mucosa than you are able to take it up, to absorb it. And last but not least, the products that were on the market in the past are also all ferrous products, Fa2+ products, and these products cause intolerance very commonly in IBD population, and also inefficacy. This was the reason why we started our first interventional trial with an IV product. This was an old drug that was available since the 70s called Ferrum Hausmann. It's an iron sucrose or iron saccharate. It's the same thing for the same name for the same thing. And what you can see here on the response curve is that on the dashed line, this is iron treatment only. 80% of the population was responding to IV iron therapy. And if you combine it with erythropoietin, it's almost 100% that responded. So we were very glad about this effect. This study was followed by a couple of review articles. I don't want to go into details. I was in the position, or I was asked to get into the position to write a couple of guidelines on the diagnosis and treatment of iron deficiency anemia in IBD. With many international co-authors, and this was basically, and we also developed a couple of other intravenous iron products such as ferric carboxymaltose, and we also developed new treatment guidelines regarding the dosing of these IV products that became more commonly used in IBD in Austria and Europe. What is the problem with ferrous iron uptake and also iron uptake from food? The main problem is that iron is absorbed mainly in the duodenum and upper jejunum, and if you take ferrous iron in a dose of 100 to 200 milligrams a day, it's only 10 milligrams to max 15 milligrams that are absorbed here. The rest of the iron passes through the small intestine and passes through the large intestine, and thereby it passes the area of inflammation. And when it passes the area of inflammation, this ferrous iron reacts to ferric iron and produces reactive oxygen species. This is something that ferric iron itself cannot do anymore. This is, I think, one of the reasons why ferric maltose is has much less side effects than ferrous iron. Side effects, you know, in IBD we have a lot of diarrhea, but in other patients like women there's a lot of constipation, but most importantly it's abdominal pain, and many think that this abdominal pain is gastric pain. Then they combine it with a PPI, which I don't think is a good idea because this reduces absorption. You need the acid to absorb iron in the duodenum. So if you combine it with PPI, don't do it. It doesn't increase efficacy and doesn't reduce symptoms. This seems to be a complicated slide, but let me lead you through that. I mentioned that iron is taken up in the duodenum, and this is a very specific process that is taken up over certain molecules. Iron is from the duodenum goes into the circulation, is bound to transferrin, and actually if you measure the amount of iron that is bound to transferrin, it's only 3 milligrams for the total body. So the iron that circulates on transferrin is minimal to the iron amount that is in the body. The total iron amount in the body is in the grams. We talk about 3 to 4 grams in an adult human. But over the transferrin, iron goes to the bone marrow, leads to erythrocyte production. Erythrocytes live for about 120 days. They get destructed in the spleen, go into macrophages, and then iron should be released back and recirculate into transferrin and being reused. It's a recycle mechanism that is very important, and it's about 20 to 25 milligrams per day that are recycling compared to this 1 to 2 milligrams that are absorbed. So the recycling is much more important than the absorption when it comes to iron metabolism. However, during inflammation, the liver builds a peptide called hepcidin. This hepcidin has certain effects. During inflammation, it blocks absorption, it blocks the release from the recycled iron, and it blocks the release from iron from the liver, and this leads to a reduced transferrin saturation, to reduce plasma iron, and to reduce iron in the circulation. I'm sorry, I'm a little bit under the weather in my voice. If I get a cough, I apologize. I can't be contagious over Zoom, but yeah.
Don't worry, Chris. We are good to go.
This is the first study actually from 1998 where ferric trimaltol was tested in humans. This is non-IBD population. You see individual improvement on ferric maltol. The important question in this study was, is it absorbed? And yes, it looks like it's nicely absorbed. Also, the ferritin increases over time. This is a study over 3 months. The same group tested absorption with different chemical formulations of ferric maltol, and they saw that all the chemical formulations they used actually are nicely absorbed. So it looks like the absorption of ferric iron does also work. We started working with ferric maltol in mice. Here we were actually interested in the toxicity of iron sulfate, which we commonly use in IBD populations, and wanted to compare this to ferric maltol. We also used a plant iron, which actually was also produced in India and came from curry leaf extract from India. And finally, we compared it to iron EDTA, which is basically the main iron you find as a supplement to food. I don't know about India, but in Europe and in the US, certain food is supplemented with iron because iron deficiency is a very important issue, and so supplementation with iron takes place. So this is a food supplement, nutritional supplement that is added to food such as cornflakes, butter, spreads, various forms. And we thought, this must be super safe. This we're interested to study. This is our product that we know, and the plant iron, we don't know how it works, and we also test. Interesting on that picture is also that iron in different formulation looks very, very different from green to orange. So iron is not iron. Iron is a very diverse metal. On this busy slide, I will lead you through. Just focus on the left column here. We added these 4 iron products to the food of mice in the so-called AOM ESS model. So mice received DSS on 4 different locations for 5 days, after 2 weeks, after 4 weeks, after 6.5 weeks, and here at the end. And DSS induces inflammation in the gut, and this inflammation you can see here with the disease activity score going up. Then you stop the DSS, the disease activity score goes down, and you add DSS, disease activity goes up, and then it goes not as much down as at the beginning because it was the second circle, and here's the third circle. On the last circle, the mice were sacrificed. One curve is going up and staying up, and these mice were very, very sick. And in these very sick mice, I need to, sorry, oops, because I have here, no, it's better. These mice were the EDTA treated mice. If you compare this now on the mean DIA, disease activity score, you see it's significantly higher in the EDTA mice. If you look into the histology, it's significantly higher, and also if you look into tumor burden, the mice received AOM at the beginning, they also have a very large amount of tumors. We were not able to really believe that. It was very unusual to us that the food supplement is so toxic. And so we used the second mouse model, the IL-10 knockout model, to replicate these data. And you see again, in the IL-10 knockout mice, the disease activity goes up and EDTA stays up and stops here because we had to take the mice out of the study because they were so sick. Again here, disease activity much higher, histology activity much higher, and tumor burden much higher. Here you see an example of EDTA-treated mice control group, and here the EDTA-treated mice, and in the IL-10 knockout mice, the same was true here. Totally destructed intestinal mucosa upon EDTA. We studied in T84 cells the tight junctions, cadherin and desmoglobin, to see what happens upon EDTA treatment, and the intestinal barrier breaks down, of course, on inflammatory parameters, inflammatory cytokines like TNF-alpha or interferon gamma, but also on EDTA itself, and the combination is most severe. This you can see also here on the FITC dextran permeability, and this is in vivo permeability on confocal, where we added EDTA in the mice, and this is also seen by electron microscopy. So EDTA is very toxic, while the other iron products, including the iron trimaltol, seem to be quite tolerable and did not cause any unexpected effect. This study was published a couple of years ago. Heiko study is the first author in that, and it should just give you a little bit of warning that food supplementation, when used by food industry, does not mean that it's really safe. Let's go to the clinical trials with ferric trimaltol, the so-called Aegis studies. These trials were done to demonstrate efficacy and safety in the IBD population. We had one trial in ulcerative colitis, one trial in Crohn's disease, and then we had a follow-up trial in both diseases for 52 weeks. This is the trial design. It was a double-blind trial. Patients were randomized one by one. It's quite a significant number of patients that were all anemic. They were not allowed to be too anemic, they had to have a hemoglobin above 9.5 because they were randomized to placebo. Our institutional review board did not allow us to randomize patients with a hemoglobin lower than that because they would be randomized to placebo. Then they were in the 12 weeks treatment phase, and then the anemia and iron status was evaluated. The primary endpoint was the change in hemoglobin between baseline and week 12. And then there was the follow-up open label trial where all patients received ferric maltol. I have to say, ferric maltol was given at a dose of 30 milligrams twice a day. You will wonder and compare this to trials with ferrous sulfate, where doses of 200 milligrams or more per day are given. And this is the result. Primary endpoint hemoglobin increase. You see a very nice change upon the active drug from average 11 to over 13. And you see also a nice response if you evaluate patients above 1 gram per deciliter. 80% of patients had an increase of over 1. 56% of patients had an increase over 2, and a significant number of patients really increased, let's say 2/3 normalized their hemoglobin. In the follow-up trial, things were as expected. This here shows you the randomization phase that I just showed you. On placebo, there was no response, and these patients were switched then to the active drug, and you see a nice increase in the response also in these patients. Again, the number of patients who normalized on the active compound is up to 92% in this IBD population. When looking into ferritin, things were replicated. Again, in the double-blind trial on placebo, no response on ferritin, and a nice increase in the active patients. But what's interesting on this graph is that you see a continuous increase of ferritin in this population for the 52-week prolongation period. So iron is continuously taken up, and we do have a positive iron balance in all these patients. Now let's switch to the adverse events. And I need to point you that this here is a 20% adverse event marker, so this would be 1/5. Of course, nasopharyngitis, these are trials in Europe, and they go over a year. It's very likely that patients feel like me today and become sick and get a cold. I don't think that is related to any of the active drugs. And you also see in the placebo group there is a significant amount of nasopharyngitis. Abdominal pain seems to be a little bit related, but not very much. We had about 8% in the placebo group versus up to 16% in the active group. The same is true for diarrhea. There's a certain amount of ulcerative colitis patients, but not Crohn's disease patients with a little worsening. But the rest of the side effects basically balance out between placebo and ferric trimaltol. Also in earlier days, we were already interested in comparing IV iron and ferrous iron. This is ferrous sulfate. These were studies from the first studies from Scandinavia. Stefan Lindgren from Sweden looked into response rate, and on the red here is the IV iron sucrose. That is basically the iron that I used in our first trial in the Annals of Internal Medicine, which I showed you at the beginning. Here, response rate of 65 to 70%, oral iron significantly less, but again, this is not the ferric maltol, this is the iron sulfate. I will get to a comparison trial between IV and ferric maltol a little bit later. If you look at the amount of patients which are anemic at the end of treatment with ferrous sulfate, it's over 40%. So the drug is not working as good as it should. We also did a comparison directly, not here with ferric sulfate, but with ferric carboxymaltose. This is the newer drug that we helped to develop. And again, you see a more significant impact drug than on the ferrous sulfate over a 12-week trial. When we compare various trials with IV and oral compounds, and you lay over the response rate of the AGIS trials that I showed you, you see actually a very nice improvement on the ferric maltol beyond the IV iron. And this was the reason why colleagues in Germany studied the comparison between oral ferric maltol and intravenous ferric carboxymaltose, which was basically the latest drug that we helped to develop in patients with IBD. This was also a randomized non-inferiority trial, and it showed significant, it didn't show any significant difference between the two products. On the first 4 weeks, there might be a little bit more improvement on the ferric carboxymaltose, but everything levels out within 10 weeks in this trial. So you can say ferric maltol is similar to IV iron treatment in this difficult to treat population. So having said that, let me summarize the data so far. Ferric maltol is effective in the treatment of iron deficiency anemia and IBD. In mild IBD, it's comparable to IV iron. It has a favorable safety profile, and last but not least, that's a little bit of a side taste here, bitter taste. Be careful with iron supplemented food. We actually recommend patients to look at food additives a lot and not to use products with food additives.
Thank you. Thank you.
I promised you to show you the indent.
Yes.
This is Vinita Karen. She's the inventor of LOHA.
Oh, yeah.
I showed you data from Aiko Estatiev. Stephanie Kulnik, unfortunately, is not on this picture. But yeah, you see that the group was proliferative also into the next generations. And I hope they will be proliferative in the future. Let me probably warm up the discussion with two cases. I brought up two cases of typical IBD cases. The first is a female, 36 years old. She has 2 children and she is vegan, so almost Indian. And she has extensive UC since 2010, recurrent mild anemia. She was on azathioprine until 2021. And in combination with infliximab. And when we give infliximab infusions, we like to add iron sucrose after the infusion. So it was basically one infusion with infliximab followed by an infusion with iron sucrose. This was done until 2018. Then she decided to get pregnant. So if you look here, this was the area of infliximab and iron sucrose. We were able to stabilize her hemoglobin and to keep her disease. When she got pregnant, she got iron deficient again and had 2 children. And she wanted to restart in 2022 with infliximab. However, she developed antibodies and was not able to tolerate this, so we switched to Entyvio, vedolizumab. This was here at this point. During the induction therapy, we also combined, this was here. And then she switched to maintenance subcutaneous Entyvio treatment. And because she didn't need a needle anymore, we added ferric maltol. And what you see on ferric maltol, a nice further increase, and the patient is completely out of anemia during this period of time. Having said that, she has a very good quality of life. She doesn't need IV infusions anymore, and she maintains her good state with subcutaneous biological treatment combined with ferric maltol. The second case is male. He's Crohn's disease using the Vienna classification. Thank you for mentioning this. I really appreciate that. The Vienna classifications, he was A1, L4, B2, meaning that he had upper GI disease, meaning small bowel disease. So he was diagnosed in 2008 with ileitis and 2 skip lesions, 50 centimeter proximal of the terminal ileum. This was followed by resection of the terminal ileum, so ileocecal resection, and proximal ileal resection. He started with Humira, adalimumab, continued with infliximab, and developed a stricture at the proximal anastomosis and received stricture plasma. This was in 2020, and if you look here at the hemoglobin at 2020, this was when his iron deficiency started. We often see patients with Crohn's disease on stricture plasters with significant bleeding and very difficult to treat. So in the years to come, he was on IV iron, and if you look at the dose, significant dose, 3 to 5 gram per year. I didn't write per year, this was 3 to 5 gram per year. This was basically total body replacement of iron. If you translate this into blood transfusions, you can say this is about 15 to 25 blood transfusions per year. 15 to 25 blood transfusions per year in the amount of iron. We used several tricks, but at the end we switched to a combination of golimumab and ustekinumab, Simponi and Stellara, at least these are the names in Austria, both subcutaneous, and added ferric maltol. And around this combination, you see a very nice increase and the stop of this anemia. The second thing which we like to do, if we reach the stricture plastic endoscopically, we do APC. This is the stricture plastic of this patient. You see here active disease. This was treated by biologicals, but you see also the bleeding. And what I like to do in these cases also use APC for coagulation. I did this twice in this patient, and thereby we solved the problem of anemia. And I think this is the point where we can start our discussion, and I'm happy to take your questions. Thank you very much.
Wow, thank you, Trish, for those wonderful insights. I really appreciate your time and your deliberation. That was really insightful and very highly informative. So I'll open up the entire panel for the question and answers. Now, before I start doing that, let me have some housekeeping rules. So good evening, friends from various centers, those who have joined with me. I would request all of you, I'll call up the name of the center, and I would request you people to restrict 1 or 2 maximum questions allowed per center, so that we can have better discussion, and we'll be also restricting our time. Also, I would request that please ask the questions through the moderators. So moderators, please unmute yourself when I call up for your center place. And in the last, we'll have some take-home messages, and then we'll have the final concluding remarks from Robert Chris. Friends, it's been really intriguing. Iron deficiency anemia, we all are aware that it's a magnificent. If you really look at the amount of the red blood cells which are synthesized or manufactured in the bone marrow, it technically boils down to almost 173 billion RBCs every day are produced. That is almost 21 times of the global population and 121 times of Indian population. That happens in bone marrow. And one of the primary things which drives that is iron. So I'll first come to our center from Behrampur, Odisha. And if we have Dr. Deepak Kumar Prushti to ask this question or two from the center. Over to you, Behrampur, Odisha. Anybody from Behrampur, Odisha? Can you unmute yourself, please? Please unmute yourself. The center has to unmute then. Please unmute. Hello? Yeah, you are audible now.
Hello. Good evening.
Good evening, sir.
Myself, Dr. Deepak Prushti from Behrampur, Odisha.
Good evening, sir. Pleasure.
We want to know about the dosage schedule of this Megaferon. How it does it affect the gastric mucosa and what is the cause?
Fantastic. So the question, Chris from Behrampur, Odisha is, what are the dosing schedules that you recommend and why it is different than the regular traditional iron preparations that we use in our day-to-day clinical practice?
Exactly.
So the dosing which we use in the studies is also the dosing which is packed, which is 30 milligrams per tablet. In the studies we used it twice a day. In clinical practice we use it typically once a day if patients are not severely anemic, and if patients are significantly anemic below 10, we also use it twice a day. I think the main difference is ferric versus ferrous, and I tried to show you that ferric iron would not induce the Fenton reaction while ferrous iron does. Having said that, of course, iron can change from the ferric to the ferrous condition, and thereby it can also harm a little bit, which you see on the adverse events, but I think it's much more safe than using ferrous iron.
Is it that ferrous part is not interfering with gastric mucosa?
Please repeat your question.
Yeah, so I'll repeat his question. He wants to know whether ferric form gets absorbed better and is not inhibited by the gastric mucosa or a presence of gastric acid. Am I right, sir?
Yes.
About the acid, I don't know, I have to say. And when they absorb, it's definitely absorbed better during inflammatory states. When hepcidin is active, ferrous iron is really badly absorbed. The absorption rate is extremely down, while ferric iron can be continuously absorbed. The absorption process of ferric iron is not perfectly understood. It's a very small molecule, and people think that it can transmigrate the mucosa without absorption using the typical iron absorption pathways.
So in fact, Chris, to elaborate a little better on that, we have been always taught in our residency days, when we were taught about iron deficiency, iron absorption, it was always told to us that in the presence of this dimethyl transferase and maybe probably some vitamin C or ascorbic acid, where ferric gets converted to ferric absorbed in the intestinal mucosa. Do you mean to say that ferric maltol, rather than following the traditional iron absorption path, bypasses the whole system and
Yes.
has an ability to get absorbed without getting even converted to ferrous form and directly get absorbed and get through the ferroporotin to the aport, means transfer it into the straight with the liver and bone marrow, right?
Yes, that is the hypothesis. It has not been proven, but it seems that in this ferric trimaltol form, iron is absorbed bypassing the MT1 and ferroportin.
Perfect. Thank you very much, sir. And thank you.
Just 1 minute. Is
Yes.
it sufficient? This 29 milligram of elemental ferric maltol is sufficient?
30 milligram is definitely sufficient, yes.
Sorry, 1 minute. Today's presentation is on this Megaferon, ferric maltol. It is prepared, I think, best for this inflammatory conditions. What about non-inflammatory conditions? Is it the same, the drop of paper with those conditions also? Number one.
Yes.
Number two, doses. Is it varies from pediatric and geriatric group? Okay.
So first question, in non-inflammatory conditions, it's also highly absorbed. We also like to use it in women. We use it in pregnant women. And in children, I don't know because I'm not a pediatrician. But I guess because of the safety, I don't see any problem also using it in children.
Right. But to extend to that, to your answer, sir, I would say that maybe probably, in a traditional non-inflammatory iron deficiency anemias, you do have other therapies as well. But this therapy can be also used, but it has a strong advantage where the traditional iron preparations, they fail. Probably, ferric maltol has an edge when you are dealing with additional inflammatory challenges. And that is generally a problem. Not only there is also a problem because of the excessive hepcidin levels. So there are problems with the iron metabolism also. And that leads to what he was trying to allude to, what we call it as Fenton reaction, where you realize that generally in iron deficiency, what we find out, that you diagnose it when the serum ferritin levels are very low. But generally when iron deficiency is associated with the anemia related to the chronic disorders, you'll always find out that the serum ferritin levels are above 100. And that tells you that this is the indication where you should not be using iron therapies to manage iron deficiency. Am I right, Chris?
I would say so, yes.
Yes. Thank you, Odisha Center. Now I'll come to Pune Center, to Dr. Gnanoba Bhaskar, a consultant physician and diabetologist from Pune, Maharashtra. Sir, your questions, please. Pune Center, please unmute yourself.
We don't have questions from audience till now, but I would like to ask one question to Professor Christopher.
Yes.
In clinical practice, how to use the hepcidin level to differentiate the anemia?
Oh, nice.
We're not measuring hepcidin. It's a very simple question. We're not measuring hepcidin in clinical settings. Hepcidin measurement has not been introduced into clinical practice because it's quite difficult to get reliant data.
Absolutely, and it is an acute phase reactant. So hepcidin levels, they get affected by so many other external factors as well. So in fact, in the research settings, yes, we do have but we have simpler tests to know, right? You can do the transferritin saturations, or even you can do the serum ferritin levels, apart from the serum iron levels that we hold them. And some laboratories will also give you the ratio of log transferritin versus log ferritin. That will also help you in differentiating and diagnosing the iron deficiency anemia and the related challenges.
Okay, and next question. Generally, we give once a day dosing, means in the fasting state or 2 hours after the food. But you said that this can be given, ferric maltol can be given twice a day. So does it affect hepcidin level, means absorption of iron, giving twice a day?
We don't know that. We designed the trials using it twice, 30 milligrams, because we were optimistic that 60 milligrams a day would be effective enough. And yes, 60 milligrams a day were very effective in the trials, as I showed you. In clinical practice, as I mentioned before, For mild anemia, we currently use it only once a day. 30 milligrams only for significant anemia, we use it twice a day. I can't tell you, whether it affects hepcidin levels because it has not been studied. Coming back to your question before, actually, what I rather use instead of hepcidin is really standard markers of inflammation, such as CRP or blood sedimentation rate, to judge the amount of inflammation and to judge the amount of iron absorption problems in these patients. When patients have high CRPs, you have to be aware that you get significant absorption problems.
Yeah, so to continue with the same question, Chris, I want to know, do you still recommend people to stay away from the calcium preparations or dairy products before administration of even ferric maltol, which is generally a traditional practice when you are prescribing an iron? This is a clear-cut instruction those are issued to the patient that please avoid taking it with food, and especially food with dairy products, and do not combine it with the oral calcium therapy.
No, we recommend taking it fasting, and we recommend, we don't recommend not taking dairy products when they're on iron treatment. Iron treatment has to be a continuous treatment for months, even years, and we would not reduce dairy products in our patients.
Thank you, Pune Center. Now I'll come to Warangal, to Dr. Chandrasekhar. Dr. Chandrasekhar from Warangal.
Yes, sir.
Please ask your question, sir.
So good evening, sir. Happy to see you on this kind of webinar, sir. Mangesh, sir. Is it audible?
Good evening, sir.
Good evening, sir. So happy to see you, sir, in this webinar. Your moderation is very excellent. And also it is very happy to hear from Professor Christoph Gash regarding this very important topic, sir. So we center, we have one question that what Professor Christoph shown in one this thing. So the study has compared with the placebo, but study is not compared with any other iron preparations one, and there is no IV comparison. So that is one question, sir. Second thing is that anemia of inflammation is different from anemia of the iron deficiency, because it is not true deficiency.
Excuse me. Some.
Yeah, sir. Christoph, sir. Professor Christoph.
I couldn't understand anything. I'm sorry.
You couldn't able to hear?
I am able to understand, Chris, about this, so I'll ask. But before that, I would request the technical team to please mute all other people, those who are talking in the background, please. Please, everybody, I am requesting you guys to keep yourself on mute when somebody is asking the question. So the question is primarily as far as the AG study is concerned that you showed. How about the comparison of this drug with the injectable preparations or IV preparations? Number two, he wants to know that most importantly, when you will say that this is the time to shift from oral preparation to an injectable preparation, is there any or something called as a treatment failure? And he wants to ask also that iron deficiency because of the chronic diseases or chronic metabolic diseases or chronic challenges, it's completely different. That we know it as an iron, the anemia of the chronic disorders, and there, is it comparable with the traditional iron deficiency anemia? So these are two questions that he wants. Am I right, sir?
Right, sir. Actually, because in this one anemia of inflammation, there is entrapment of this iron by the macrophages, so there is no true deficiency of the iron, sir. So yes. You are nicely narrated the question, sir.
So that is the place where probably the role of hepcidin and its interference with ferroportin comes into the picture.
Yes, sir. Yes.
So, most of the iron gets deposited in the reticuloendothelial system and maybe probably the intestinal cells, and it is excreted unabsorbed. So, over to you, Chris.
So, first question, comparison to IV. There is only this single trial that I showed you from Germany that compared ferric carboxymaltose to ferric trimaltol. So, injectable or intravenous treatment versus oral treatment, which was very comparable. For clinical practice, if patients are very sick and have severe anemia and you have IV treatment available, I would use IV. And if the patient is also able to come to the center and get the infusion. I know India is a large country and patients live apart from doctors who are able to give infusions. So I think for patients who have a hard time to get an infusion, I think it's a very good option to get this well-absorbed drug orally instead of other oral compounds, ferrous compounds. So this is our clinical practice at LOHA is severe anemia is treated IV. On continuous patients, I showed you subcutaneous biologicals and stuff like that. We switched to oral iron with ferric trimaltol. About the question, anemia of chronic disease, how much does it impair the activity of that compound? I'm sure it does impair the activity of that compound. We do see this clinically. The sicker the patient is, the less response you have. However, the studies showed that also patients with active disease were partially responding to that compound. So I would say it's better than nothing.
And how about your experience, Chris, in the patients, those who have got chronic anemia because of the disease like diabetes or CKD, apart from the inflammatory disease? How will you place this molecule in the management of anemia of chronic diseases, which generally present
I think it's a very good molecule for maintenance, for long-term treatment, where patients don't want to get infusions.
Thank you very much, Chris. Thank you, Warangal Center. Thank you very much for being there. Now I'll move on to now Jorhat. Dr. Sujay Barua from Jorhat. Sir, question from your place. Do we have anybody from Jorhat?
Jorhat is muted.
Yeah.
Hello.
Yeah, audible. Yeah.
Okay. Actually, from Jorhat, I would like to ask, in Assam, we used to get anemic patients, very severe anemia, 6.5, 6, 7, in that range. Whether this ferric maltose, you have any studies related to very low iron which respond to ferric maltose?
Yeah, so question is that is this molecule useful to manage very severe anemias where HbE hemoglobin is less than maybe probably 7?
Yes, so we do in clinical practice, I mentioned before, we do treat patients with severe anemia intravenously to make sure also the compliance issue is not there. The patient is getting iron 100% intravenously. If this is not possible for various reasons, I do think this is a very good compound. I would use it twice a day in these patients and try to control the response in shorter periods of time to make sure that the absorption is appropriate and the response is appropriate.
So in fact, shall we draw a conclusion that generally if the anemia is very severe, ideal to start with the parenteral iron therapy first? Correct?
I think it's, if you want to be on the safe side and if the patient wants to be on the safe side, we use IV for number one, for compliance, number two, for there is no issue about absorption. The drug is 100% in the body, and we don't need to worry about whether it gets into the body or not. The bioavailability of IV is 100%. The bioavailability of oral drugs is never 100%. The bioavailability of ferric trimaltol is significantly better compared to ferrous products, so I would prefer that product over others in the IBD population. But yes, in Austria, patients are living very close to doctors. I don't know how many gastroenterologists or how many doctors per capita is in India. In Austria, we are one of the countries with the highest number of doctors per capita. And so patients with significant anemia always get IV iron therapy.
Fortunately, in India, we do have what that lot of disproportion between the patient and the doctor. So, and we are working on it, and I'm more than sure that in coming time we are going to do it much better, honestly speaking. So, there's an age study that you show, where their population people were involved with severe anemia versus mild anemia, and was there any difference in the response or the delay in the response?
So I mentioned that in the AGIS studies we were not allowed to include patients with severe anemia because they were randomized to placebo. Our ethics committee would have considered it unethical to randomize somebody with a hemoglobin of 7 to placebo. So this is withholding of active therapy. This is unethical. So this is why patients had a hemoglobin when they were in the AGIS studies.
Oh, thank you. I'll come to Dr. Madhurika Agarwal from Jaipur. Thank you for being there, ma'am. It's a pleasure and honor to see you. Please unmute yourself and then ask your question from your center, please. You need to unmute. Yeah.
Hello. First of all, I would like to congratulate Dr. for such a nice and elaborating lecture on parents are housing alters. My question is, sometimes the ferritin levels are unreliable. In that case, what is the best diagnostic tool to diagnose anemia?
There's a lot of background noise, but what
I understood that ferritin is unreliable, and what is the best diagnostic tool to diagnose anemia?
Is that
that is correct. Ferritin is on various levels unreliable. However, a low ferritin is always low. Okay, high ferritins are not necessarily showing that there's high iron content in the body. It can be due to inflammation, high ferritins, but a low ferritin is a very good parameter. So if you have ferritins below 30, and this is the cutoff which we are using, you can 30 milligrams, though if you use higher doses, you won't find significantly more adverse effects. So I think it's a safe drug, but
Thank you, sir. Thank you for taking your busy schedule, taking up that some time and attending the program. Once again, thank you each and everyone. Kindly break for the dinner, sir. Before that, I request Chandrasekhar sir to come to small felicitation. Just 1 minute, sir. Please, we request.
అయిపోయింది లైవ్ ఓకే